Hi everyone, we are a group of students from Stanford University who are studying general microbiology. My name is Peihan and today the topic that me and my groupmates are going to present is Microbes are the future, Pharmaceutics. This group consists of Zijun, Jian, Ming, and Junki. So, we will be introducing how microbes such as the E. coli are used in the pharmaceutical industry and how these microbes can help humans in the future. Pharmaceutics is a branch of pharmacy that deals with the formulation, manufacturing evaluation, and packaging of pharmaceutical dosage forms. And microorganisms or microbes are widely used in the pharmaceutical. Now, we will first look into the taxonomy of the E. coli. The E. coli fits into the domain and kingdom of bacteria because they are unicellular microorganisms. Next, E. coli fits into the phylum. Next, we will move on to the structure of E. coli. The E. coli is a bacillus where it is rod-shaped. It has a dimension of about 1.5 micrometer long and 0.5 micrometer wide, and it is alright. So now we'll move on to the characteristic of E. coli. First, E. coli is a gram-negative bacteria because its cell wall is composed of a thin peptidoglycan layer. Next, alright. So now we will move on to the growth conditions of E. coli. E. coli grows optimally at 37 degrees Celsius under aerobic conditions. However, there are some laboratory strains that can multiply. However, there are some virulent strains like O157H7 that can cause diseases such as UTI, Crohn's disease, and hemorrhagic colitis. The common signs and symptoms include abdominal cramps.
Back in 1978, a significant milestone was achieved at City of Hope when scientists successfully produced recombinant human insulin in collaboration with Genotype. This breakthrough marked a pivotal moment in the field of biotechnology. Subsequently, Eli Lilly and Genotype. The initial approach involved the cultivation of separate A and B subunits of insulin in E. coli as inclusion bodies. These chains were then purified individually and chemically combined, followed by further purification steps. This method, known as the two.
Before we talk about the mechanism of action, recombinant DNA is a technology developed to insert a human gene into a genetic material of a common bacteria. This recombinant microorganism can now produce the protein encoded by the human gene. So in terms of this recombinant insulin from E. coli, the mechanism of action exerted is start from production. So the scientists built the human insulin gene in a laboratory, then they remove a loop of bacterial DNA known as plasmid and insert the human insulin gene into the plasmid. Researchers return the plasmid to the bacteria. The recombinant bacteria is put in large fermentation tanks after the researchers return the plasmid to the bacteria. Then, the recombinant bacteria use the gene to begin producing human insulin. Then the scientists harvest the insulin from the bacteria and purify the substance for use as a medicine for people. So we are done with production. Then, the recombinant insulin is typically administered via subcutaneous injection. After the injection, the insulin analogs are recognized by the insulin receptor and bind to it. Then, the insulin is absorbed into the bloodstream, and the lymphatic system also plays a role in transport. The absorption of human insulin into the bloodstream is inconsistent and varies around 15% within an individual and 30% between the patients. Most of this variability of insulin absorption is correlated to blood flow differences at the different various sites of injection, like injection from the abdomen, thigh, and so on. Once the insulin is absorbed, it regulates various metabolic processes such as protein synthesis and inhibiting the breakdown of stored glucose and fat. Moving on to the example, the first recombinant insulin approved in 1982 is the Humulin. However, these subcutaneously injected recombinant insulin usually have a slow onset with peak plasma concentration after 2 hours of injection and longer duration of action that lasts for 6 to 8 hours. Hence, fast-acting insulin analog was developed. So the first fast-acting insulin developed was by Eli Lilly and its name is Lispro. A synthetic cDNA encoding for Lis B28 and Pro B29 human proinsulin was expressed in E. coli for commercial production of insulin Lispro. So the insulin Lispro was excised proteolytically from the proinsulin by treating with trypsin and carboxypeptidase. And then the insulin glargine is one of such long-acting insulin analog. So glargine was produced as proinsulin and expressed as E. coli as well. It was finally formulated at pH 4 in soluble form. However, after subcutaneous administration, it precipitated due to neutral pH in the subcutaneous tissue. Resolubilization of insulin occurs slowly, resulting in longer duration for its release in the blood. And moving on to the impacts on the humans. So the first one is selective action. Insulin has a number of effects in addition to carbohydrate metabolism. Some of these effects depends on the cell or tissue type studied. Therefore, selectivity of an analog can be defined as the selectivity action towards a certain tissue or to a certain effect. The second one is the stability can be increased. So the insulin is not a stable chemical entity actually. The insulin decomposition is mainly due to two categories of chemical reactions, which is hydrolysis and intermolecular transformation leading to covalent insulin dimers. The identification of the residues undergoing chemical changes during storage allows the designing insulin analogs with improved stability. So the advantage of such analogs will be prolonged shelf life and more convenient storage conditions. Improved stability is also essential for pump usage. And then moving on to the last impact, which is the variability of insulin responsiveness will be lessened. So there are two explanations for the variability of insulin responsiveness. The first one is the pharmacokinetic variability can result from variation in insulin absorption, leading to different plasma concentration of insulin after injection of the same doses. The second one is the pharmacodynamic variability can be caused by the variation in insulin action, causing different metabolic effects by similar plasma insulin concentrations. So for example, after the insulin Lispro was injected, a decreased variability in serum insulin concentration compared with regular human insulin has been shown.
A versatile bacterium has shown great potential in the pharmaceutical industry. Well, my discussion today is based on scientifically sound assumptions and ongoing research. The abstract sets the stage for exploration, highlighting the exciting possibilities and challenges that lie ahead. Expanding biomanufacturing capabilities is a crucial aspect. We assume that E. coli will continue to serve as a reliable host organism for producing pharmaceutical compounds for the future timeline ahead. One assumption is that the protocols used in culturing E. coli could be scaled for industrial usage. Ongoing research by pharmaceutical companies and startup labs have already generated some protocols to be used in larger scale production lines. Efficiently optimizing metabolic pathways is crucial for enhancing yield, purity, and productivity in biomanufacturing processes. Scientists and researchers are working tirelessly to fine tune these pathways, allowing E. coli to produce complex molecules more efficiently. Additionally, addressing endotoxin-related concerns is essential to ensure the safety of our final pharmaceutical product. Large-scale production is another challenge that needs to be overcome. Developing robust fermentation strategies is necessary to scale up E. coli-based production, and researchers are also exploring various techniques and technologies to achieve this, such as improved bioreactor designs and innovative fermentation methods. Now, let's shift our focus to the exciting realm of novel therapeutic approaches. E. coli-based systems are expected to contribute to the development of innovative therapeutic modalities to actually allow us to better treat things and treatments for certain diseases that we never once had. One assumption is that E. coli can be engineered to produce compact biologics like monoclonal antibodies, vaccines, and protein therapeutics. Ongoing research suggests that E. coli can be modified to produce these biologics efficiently and quite low cost. However, challenges exist in this domain as well. One of the main hurdles is protein folding, stability, and post-translational modifications. E. coli may not possess the necessary cellular machinery to fold and modify complex proteins correctly. Scientists are actively working on engineering E. coli strains that can overcome these limitations, ensuring proper folding and stability of therapeutic proteins. Another challenge lies in the proper glycosylation of therapeutic proteins. Glycosylation plays a crucial role in their function and stability. Researchers are exploring different strategies that allow E. coli to perform glycosylation processes effectively, opening up new avenues for therapeutic development.
Alright, now let's have a small recap, shall we? First of all, we discussed that E. coli plays a big impact on humans, such as aiding in our digestion and protects us from harmful microbes. Although, on the other hand, where virulent strains like O157H7 cause diseases such as urinary tract infection, hemorrhagic colitis, and Crohn's disease. We also discussed how recombinant insulin can be produced with two strategies, known as two-chain combination and enzymatic cleavage, by inserting a human insulin gene into an E. coli plasmid. Subcutaneous injection is often used as a technique to insert insulin into the human bloodstream. As it enters our bloodstream, insulin acts as a hormone and binds to our insulin receptors, triggering a series of intracellular signaling events. This activates our glucose transporter proteins to facilitate the glucose from the bloodstream into the cells. The glucose is now stored as glycogen and can be converted back for energy production. With that being said, there are several types of insulin invented for different objectives. Lispro and glucagon was mentioned as an example for the fast-acting insulin analog developed by Eli Lilly and Avantis Pharmaceuticals respectively, whereby another type of insulin analog are long-acting insulin analogs known as insulin glargine. As until today, with the ongoing research, we mentioned that E. coli has shown great potential in the pharmaceutical industry, and we assume that it will continue to serve as a reliable host organism for pharmaceutical compounds production. With that, culturing E. coli could be scaled for industrial usage, as some companies have already generated protocols for larger scales. Another assumption is that E. coli can be engineered to produce complex biologics, such as vaccines and monoclonal antibodies for future diseases. However, there are several challenges that we have to overcome in terms of efficiency, such as efficiently optimizing metabolic pathways to enhance yield, purity, and productivity. In addition, addressing endotoxin-related concerns and developing robust culturing protocols for industrial usage is also part of the challenges. Other than that, E. coli has a limitation where it may not possess the necessary cellular machinery to fold and modify complex proteins correctly for post-translational modifications. After conducting research on this topic, we as a team realized that even though E. coli is a simple bacterium that can be commonly found, it is impressive how versatile and useful for us humans to leverage it to our favor. We hope that someday in the future, E. coli will be the key factor to a certain disease or even in a pandemic. Even if it does happen, thank you, that's all for today.